right, everybody, let's talk about bacterial envelopes. An envelope, when we use the word to describe bacteria, refers to the arrangement of the cell membrane or membranes and the cell wall, because bacteria do have a cell wall. Now, there are two terms that we use to describe these two arrangements, and those are referred to as gram-positive and gram-negative. Gram-positive cells have a cell membrane, a regular plasma membrane with all its regular functions, and they have a very thick cell wall. So if we're going to draw them really simply, this is kind of how I draw it. Cell membrane, cell wall. That's gram-positives. Very, very simple. Gram-negatives have a very slight difference. They do have a regular plasma membrane. They do have a cell wall, only their cell wall is really thin, and they have an additional outer membrane. So they have a cell wall sandwiched between two cell membranes. Now the space in between the membranes here is called the periplasm. And there are a few other differences between our gram-positive and gram-negative cells. Gram-positives have these molecules called teichoic and lipoteichoic acid. And gram-negatives have these structures called lipopolysaccharides, which gets shortened, because that's a mouthful, to LPS. So that's how we just generally draw these two, gram-positive and gram-negatives. And it's important that you know what all of these different pieces mean. Bacterial cell walls are made up of a particular structure called peptidoglycan. Only bacteria use peptidoglycan in their cell wall. No fungi, no plants use this, so it's unique just to them. And as you can tell from its name, peptido, it's going to be made up of protein and glycan, sugar. So proteins and sugars. There are two different sugars that get linked together. They're named N-acetylmuramic acid and N-acetylglucosamine. You don't need to know that crazy long name. Just know that it's NAG, N-acetylglucosamine, and NAM, N-acetylmuramic acid. And those get linked together in an alternating pattern. Dark green, light green, dark green, light green here. And they make these long chains of NAG and NAM linkages. Each of these long chains get linked together by peptide crosslinks. Right? So the glycan part refers to the NAG and NAM, these alternating sugars, and the peptido part refers to the peptide crosslinks. This gives us a blown up view of a single linkage of NAG and NAM. Here's the NAG, here's the NAM. And our peptide chain coming off of that NAM subunit. Looking at this part that I've circled here, those peptide chains get bound to each other, and that's what provides peptidoglycan with a firm link. So I think of this as a raft. If you were making a log raft, the NAG and NAM long chains would be like the logs of your raft. But if you just have logs together, with nothing to tie them, they're not going to stay afloat. They're going to go every which way. So you have to tie those logs together with some sort of rope. And that rope for peptidoglycan would be the peptide crosslinks. Those peptide crosslinks hold the NAG and NAM together so that the cell wall doesn't just fall apart. The process of peptidoglycan synthesis is pretty involved. But there are a few parts of it that it's good for you to know, because these are often parts that are targeted by antibiotics. So a lot of the antibiotics that you take to treat a bacterial infection actually target cell wall synthesis. In our first step, that NAM is synthesized in the cytoplasm, and it's linked to a UDP there at the plasma membrane. This gets linked to a particular protein called bacterial, and after that, the NAG gets added to it. So we have our peptide, and now we have our two sugars. This is all in the cytoplasm of the cell, so this is on the inside. To get this to the outside where the cell wall is, this whole protein, this whole bacterial protein flips over so that now you can see there's the NAG and NAM right up here at the top outside of the cell. This NAG and NAM gets bound to other NAG and NAM parts through the action of a very, very important enzyme called a transpeptidase. Transpeptidase is what gets targeted by things like penicillin. This transpeptidase, it carries out that cross-linkage between our two peptide bridges there. Thus, if transpeptidase is interfered with, we don't get peptide crosslinks formed. No peptide crosslinks, and our cell wall just falls apart. Now back on the very first slide, I drew a beautiful diagram of a gram-positive cell. I'm writing gram-positive up here in purple because when we stain bacteria using the gram stain in lab, gram-positive cells stain purple. So remember purple for positive. Gram-positive cells, remember, have a cell membrane, which we can see right here, and they have a thick cell wall. Now their cell wall is thick because it is made up of multiple layers of peptidoglycan, which we can shorten to PTG. So we can look and count maybe 1, 2, 3, 4, 5, 6 layers of peptidoglycan in this cell wall. Embedded in the cell wall and cell membrane are also a number of integral membrane proteins on the cell wall. And in purple here is that teichoic acid that I told you about at the beginning. The image on the left points out where you can see these things in an electron micrograph. This darker line that you can see here is actually the plasma membrane of the cell. The next area out, which I'm kind of shading in blue, that's the periplasm. Gram-positive cells do have a periplasm. It just is a little bit different from gram-negative. And then on the very outside, this much thicker layer here is the thick gram-positive multi-layered peptidoglycan. Here we have our gram-negative bacteria. I'm writing this one in red because in the lab, when we do a gram stain, they stain red. Looking at this diagram, we see the inner plasma membrane here, the outer membrane on top, and importantly, here is our very, very thin layer of peptidoglycan. Where our gram-positive bacteria had multiple layers of peptidoglycan, our gram-negative have just a single layer of peptidoglycan. That is why their cell wall is so thin. Here again is the periplasm, this space in between the cell membranes, which includes in our gram-negative bacteria the cell wall. On the left again is our electron micrograph. This line here is our inner cell membrane. Here in blue is the periplasm. We have our very thin layer of peptidoglycan just to the outside of that, and we top the whole thing off with our outer membrane. Embedded within that outer membrane is the lipopolysaccharide that I mentioned earlier. And this is only found on gram-negative. Gram-positive cells do not have lipopolysaccharide. What is lipopolysaccharide? If you think about what's in its name, there's lipo and there's polysaccharide. Lipo referring to lipid, polysaccharide referring to sugars. This is going to be a combination of a lipid and a sugar. Lipopolysaccharide, we also just call LPS, again, because like peptidoglycan, no one wants to write or say lipopolysaccharide repeatedly. The lipid portion is made up of what's called lipid A. This lipid A is what anchors LPS into the outer membrane. And this is also the toxic component of LPS. When you have a bacterial infection and you get a fever, if you've been infected with a gram-negative bacteria, it's usually the LPS. Bound to the lipid is our large sugar portion. All of this is a sugar portion. There's what's called the core polysaccharide, and this is usually conserved among a particular genus. And then the outer part, which is kind of this orangey color up top, is the O side chain. And this one will be more specific at the species level. One more time, LPS is only on gram-negative cells. The final external structure that we're going to talk about is known as capsule. Capsule is a thick polysaccharide, lots of sugars, matrix that will sit outside of the last layer of the cell. For gram-positive cells, that would mean outside of the cell wall. For gram-negative cells, that would mean outside of the outer membrane. Capsules are very, very thick. They are highly structured, and they provide a number of benefits. One of the first is that they can prevent dehydration. A second is that in the body, they can prevent the bacteria from being recognized by the immune system. This picture shows us how thick capsules can be. The dark spots that you see here are two cells, so a pair of diplococci, and the outer layer here is the capsule. You can see that the capsule itself is about twice as thick as the bacteria within it. Imagine the amount of protection that a cell can have due to this very, very thick and hardy polysaccharide matrix that's surrounding it and protecting it from the environment. Let's do a rapid review. Gram-positive cells stain purple when we do a gram stain. They have a plasma membrane, a thick cell wall made up of multiple layers of peptidoglycan, and teichoic acids embedded within that cell wall. Gram-negative cells stain red in a gram stain. They have a plasma membrane, a thin cell wall that's made up of only a single layer of peptidoglycan, and an outer membrane. And embedded within that outer membrane is lipopolysaccharide, also called LPS, which only is found in gram-negative cells. That's it. That's our bacterial envelopes and the structures that you will find on our two different types of bacterial cells, gram-positives and gram-negatives.